Hello everyone. So in this second part, let's talk about phase 2. Let's begin our discussion with phase 2. Okay. So what happened in phase 1? It was converted to more polar compounds. In phase 2, usually with the help of conjugation, we convert the phase 1 intermediates into more water soluble, or you can say hydrophilic compounds, which will be therefore excreted in urine or bile. So conjugation, there are various ways of doing conjugation. So conjugation, we can conjugate with the help of glucuronic acid. So there's glucuronidation, I mean the Z that we used here, right? The Z that we used here, that can be glucuronic acid, that can be anything, that can be active sulfate, that can be glutathione or whatever. So glucuronidation is one way. Another way is I should say sulfation. Okay. Another way is with the help of GSH. Okay. So conjugation can be of these three types mentioned in Harper. So few points about these are glucuronidation. This is the most frequent conjugation. So first point, this is the most frequent conjugation. Okay. Now what is the donor? The donor is UDP glucuronic acid is the donor of glucuronic acid. This is the donor, right? It's similar to bilirubin, right? From heme we have from heme, it with the help of heme oxygenase we get biliverdin, then biliverdin reductase, we get unconjugated bilirubin, it goes to liver, then with UDP glucuronyl transferase. So same, similar here also we have the enzyme UDP glucuronyl transferase. I mean there can be many glucuronyl transferases, so like that, glucuronyl transferases, right? So this is present in ER and cytosol, right? So what are the xenobiotics which favor which choose this glucuronidation pathway, I should say, which goes through this glucuronidation pathway in the as the conjugation pathway in phase 2 of its metabolism. Some examples given in Harper are 2-acetyl aminofluorine, which is a carcinogen, aniline, obviously if I remember the formula, benzoic acid, then tranquilizers like meprobamate, right, phenol, then we have phenol, then steroid hormones. Okay. And these basically, these glucuronides, let me name it G, these attach with oxygen or nitrogen or sulfur atoms of the substrate groups, right? So Glucuronidation, done. Next we move on to sulfation. Okay, sulfation. So what are the compounds? Let me write them. They are, or the xenobiotics, they are alcohol. So let me write okay, let me write alcohol only. Alcohol, arylamines, phenols. Okay, so phenols can choose glucuronidation as well as sulfation, as we can see here. So phenols, right? So what is the, I mean, what is, who is the donor here? The sulfate donor here is PAPS, which is or which is also known as the active sulfate. This is the donor. What is PAPS? PAPS is adenosine 3' phosphate 5' phosphosulfate, right? So I'm not writing all that. It's also known as active sulfate, right? This is a sulfate donor, and the enzyme is obviously uh, sulfotransferases, right? Okay, then we have our GSH, right? Okay, so GSH, it acts on what? It acts on, so the xenobiotics are mainly electrophilic compounds. Okay, that's it. So obviously the thing here is donor, whatever, it's glutathione, right? What is glutathione? It's gamma glutamyl cystinyl glycine, right? Cystinyl glycine, it's found where? It's okay, so yeah, this is glutathione. So what's the reaction? The reaction basically is, if R is a, you know, electrophilic xenobiotic GSH, we have RSG, right? RSG, this is basically a conjugate, sulfur conjugate. What's the enzyme? The enzyme is glutathione S transferase. So I'm just writing GST, right? Glutathione S transferase. It's present where? It's present in the liver. That's great. It's present in the liver, and there are interesting things about it. That is, it has 4 cytosolic plus 2 microsomal membrane bound classes, right? It also has a structurally distinct, structurally, you can say structurally distinct, I would say kappa class, which is found in mitochondria plus peroxisomes, right? Mito plus peroxy. This GSH is a GST, sorry, glutathione S transferase, can be homo or heterodimers, and they are made up of greater than equal to 7 different types of subunits. Subunits, right? Subunit types or subunits, whatever. The different subunits are induced by different xenobiotics, obviously. Okay, so now this GST, this activity is upregulated. So what should I write? Activity upregulated in many tumors. So activity upregulated in many tumors. We saw what? We saw procarcinogen, phase 1, biologically active, right? I mean, any procarcinogen or whatever, any xenobiotic which is procarcinogen, which becomes active after, this thing, phase 2. So if that means they will cause cancer, right? So, or you can say that they will, they can lead to chemotherapy resistance, you can say, which will lead to chemo, because they will form a new type of thing, chemotherapy resistance, right? Okay. Now this GST also binds with not only xenobiotics, they bind with bilirubin, steroid hormones, carcinogens like ligands and substrates. So they also bind with ligandines, I want to say. What I mean is that they also they bind this glutathione transferase, bind with ligandines as well. Done. So this is done, right? And you can also say that in the liver, right, we have the liver and we have the kidney, right? We have these two things. We have the liver and we have the kidney. Right. So what basically happens is that RSG, which is if it's formed in the liver, this goes out of the liver and forms cysteine sulfur conjugate. How? What's the enzyme here? Let's mark it *1. We'll write the enzyme later. It goes to the kidney and then what happens? N-acetylation happens in kidney. N-acetyl, it's difficult to write here. N-acetylation, right? We get what? We get mercapturic acid. Okay, mercapturic acid is what? Basically N-acetyl cysteine is conjugate, this same thing, N-acetyl, we just write this thing here, N-acetyl cysteine sulfur conjugate, right? And this is excreted in urine. Okay, now what is this star 1? What are the enzymes acting there? It's gamma glutamyl transpeptidase and dipeptidase. These are extracellular enzymes. Extracellular. Okay, these are extracellular. Cool. So GSH, apart from the this conjugation, they also help in many other things. You have learned it, right? It maintains the sulfhydryl SH groups of enzymes in reduced state, so it acts as intracellular reductant and antioxidant. You have also read this in Meester cycle, amino acid transport, right? It's where it's present in plasma membrane of renal tubular cells and bile ductule cells. It's present in endoplasmic reticulum of hepatocytes, right? This gamma glutamyl transpeptidase GGT, it's also an early indication of liver damage, right, which is released into blood in hepatobiliary disease, right? So as I said, phase 2, it's about conjugation, usually, mostly, but it's also about acetylation, right? It's also about acetylation and methylation, right? We learned about conjugation, so you can say this is number A, this is number B, and this is number C. Great. Acetylation, again, same thing, SAM to SA, whatever, we have learned it everywhere. We have X here plus, sorry, not acetylation, that will be methylation. X here plus we have again, yeah, same thing only, X plus acetyl CoA, we get acetyl X, acetyl X plus CoA. So yeah, methyl donor is acetyl CoA, sorry, acetyl donor. Again here methyl donor, X plus S adenosyl methionine, SAM, we have methyl X, same thing, plus S adenosine homocysteine, SA, SAM to SA. Again the enzyme will be acetyl transferases present in liver cytosol mostly, and here it will be methyl transferases. Okay, so yeah, and this acetyl transferases, if we have polymorphism, this will lead to again slow or fast acetylators, so slow or fast metabolism, similar to previous explanations, right? So this slow thing will lead to isoniacid toxicity. Why isoniacid toxicity? I mean, why? This isoniacid, what is this? This isoniacid is a drug which is, it's used, it's a bacteriostatic drug, basically. It's used to treat TB, right? So this isoniacid, if I say, this uses acetylation as phase 2, as its phase 2 reaction. So it is a treatment for TB, and uses acetylation. So that's slow, remain system toxicity, same, just same, nothing different. So we are done with this. We are done with phase 2 of metabolism and all whatnot. So we are only left with, xenobiotics toxicity, right? So let's move on to our last broad topic, xenobiotics toxicity. Right. So xenobiotics toxicity. So there are 3 types of toxicity. So let me write them first. We have cytotoxicity, we have immunological toxicity, so I should say immunotoxicity, we have some carcinogenic, effects, right. So what is cytotoxicity? Covalent bonding, right. Let me write this in red. Covalent bonding of xenobiotic metabolites, so we have xenobiotic here, to macromolecules like DNA, RNA, protein. This can lead to, this can lead to what? This can lead to cell injury, right. Now this is cytotoxicity, cell injury, and therefore if this is severe enough, this will lead to cell death, right. Now in response to DNA damage, DNA damage obviously will be here, so DNA repair mechanisms will be activated. So obviously DNA repair mechanisms, they use ADP ribose in that, and ADP ribose, the source of ADP ribose is NAD. So in response to severe DNA damage, there is DNA repair, and hence there is severe depletion or considerable depletion of NAD. NAD depletion, this leads to impaired, I should say ATP impairment. This will again lead to cell death. That's it. That's very simple. Okay. Next we move on to immunotoxicity, right. So what happens in immunotoxicity? This xenobiotic acts as a hapten, right. Xenobiotic acts as a hapten. What is a hapten? A hapten is a small molecule which, when combined with a protein, can elicit the production of antibodies, right. A molecule here, xenobiotic, will be called a hapten, is a hapten because it binds to a protein, a carrier protein, a large carrier protein, and it elicits the production of antibodies which bind specifically to the protein. Sometimes it doesn't, then the problem happens. Okay, so when this reactive metabolite of a xenobiotic may bind to a protein acting as a hapten and altering its antigenicity and antigenicity. So X binds to a protein, therefore antigenicity is altered, right? Antigenicity is altered. So on its own it will not stimulate antibody production, but does so when bound to a protein. So the resultant antibodies react not only with the modified protein but also with the unmodified protein. So the resultant antibodies will react with protein modified as well as protein unmodified, and this protein unmodified one will lead to autoimmune disease, because we didn't want to destroy that protein, that antigen or whatever. We didn't want to destroy the protein, but antibodies, they attacked that protein. So yeah, it's autoimmune disease. It can lead to autoimmune disease, right? The third one is carcinogenic. So a xenobiotic, when reacted with DNA, it leads to chemical carcinogenesis, right? Some chemicals like what should I say, benzoalpha pyrene, right? So cytochrome P450 in endoplasmic reticulum, this activates benzo alpha pyrene, which is an indirect carcinogen, because it needs, this is activation, right? Activation, not induction or whatever, it's activation. So therefore it needs to activate this for this to become a carcinogen. So carcinogenic, cytochrome P450 is required, right? So the activities of the xenobiotic metabolizing enzymes present in the ER help to determine whether such compounds become carcinogenic or are detoxified. How it will be detoxified, right? The enzyme epoxide hydrolase in the membranes of ER can have protection against some carcinogens, like what? Let's talk something a little bit different. For example, let's talk about protection. We have here a pro-carcinogen, right? A pro-carcinogen. This reacts with cytochrome P450. We get epoxides. What are epoxides? Epoxides are compounds with this group. These are epoxides, right? We get epoxides. Epoxides are what? Epoxides are highly reactive and they are mutagenic, or I would say carcinogenic, right? Now these epoxides, they react with water and the enzyme epoxide, obviously hydrolase, pretty obvious, epoxide hydrolase, and we get what? We get dihydrodiol, the ring breaks, we get this is what? This is dihydrodiol. This dihydrodiol, this is much less reactive. Okay, this is much less reactive. So yeah, pretty much that's it. Epoxide hydrolase, this is just present in I should say ER membranes. So yeah, let's recapitulate a bit, just a little bit. What we read about phase 2. So phase 2 is conjugation, acetylation and methylation. Conjugation is again of three types: glucuronidation, sulfation and with GSH. Most frequent is glucuronidation and all. We learned about all these things. And then acetylation, methylation, then we learned about toxicity, cytotoxicity, immunotoxicity and carcinogenic effects. All these things have protective function. We learned about the protective function, and that's it. Hope you liked these two videos on xenobiotics. Thanks for watching.